Good morning. I will be giving an update today on the regulatory science program that we have for generic drugs that we've established under GDUFA. So as many of you may already be aware, the generic drug user fee amendments, known as GDUFA, were passed in July of 2012 to speed access to safe and effective generic drugs to the public. The amendments require user fees to supplement the costs of reviewing generic drug applications. But in addition, it provides resources, including funding for our regulatory research program. The regulatory science program supports access to generic drugs in all product categories, particularly products that have little to no generic drug availability. And we do this by development of new tools to evaluate drug equivalence and support generic drug development. Tools such as simulation tools to predict drug absorption, development of advanced analytical methods for product characterization, and investigation of in vitro methods to predict in vivo performance. Overall goals of our research program are to enhance access to generic versions, especially for complex drug products. Traditional bioequivalence methods may not be sufficient or sometimes may not be feasible for complex drug products. So we're really looking into investigating alternative approaches, such as in vitro approaches, to demonstrate bioequivalence. Our research program also seeks to identify issues that need to be addressed during pharmaceutical generic drug development. And we want to provide characterization methods and performance tests that will be needed for in vitro bioequivalence approaches. So since the start of our regulatory science program in 2013, we've now awarded over 100 extramural grants and contracts to collaborators out in academia and industry. We also work with other FDA laboratories as well as other government agencies. And the regulatory science program is implemented by the Office of Research and Standards, which is a sub-office of the Office of Generic Drugs. The table below shows the breakdown of the number of new projects and ongoing projects that we funded each fiscal year. And many of our projects are on multi-year timelines, and they require multi-year funding. So we do fund a substantial number of ongoing projects each year. And we also have a number of projects that we continue to provide administrative and scientific support for, but they are on low-cost extensions. So actually that number is not included on this table. The regulatory science program has 5 main priority areas, which are listed here. I've also indicated the number of extramural projects that we've awarded for each area. We have a heavy emphasis on topics 2 and 3, equivalence of complex drug products and equivalence of locally acting drug products. These are also areas that are emphasized in the GDUFA 2 commitment letter. So just in the past fiscal year, the impact from our regulatory science research program has contributed to the development of 22 new product-specific guidances and the revision of 19 product-specific guidances. In the past year, our staff has given over 100 external posters at various scientific conferences. We've given over 30 external talks, and we've published over 50 manuscripts in different scientific journals. So I'd like to highlight just a few major outcomes from just this past year. This study describes for the first time the use of open flow microperfusion as a dermal pharmacokinetic approach to evaluate topical bioequivalence. So typically for topical drug products, we recommend a clinical endpoint study, but that type of study may not be feasible or may be a huge challenge for a huge subset of topical products. So methods such as this could potentially be used to demonstrate or support the demonstration of bioequivalence. Here, we insert a probe into the dermal tissue, and we're able to collect interstitial fluid continuously for up to 48 hours. We're able to do this because the pump is connected to the probe is connected to a wearable pump, and this allows for subject mobility during the entire sampling period. In this study, we tested this method on 20 healthy volunteers. For the reference product, we used Zovarac's Acyclovir Cream, manufactured in the US. The test product, we used an Austrian generic. which has a different composition than that of the reference product. And the results of the study validated this method in that it showed accuracy, reproducibility, and sensitivity in being able to detect formulation differences between test and reference. Another research highlight from last year comes from our research on anti-epileptic drugs. Back in 2007, the American Epilepsy Society issued a position statement opposing generic drug substitution of anti-epileptic drugs until data from a valid, controlled, prospective clinical trial in people with epilepsy was conducted, studying different formulations manufactured by different manufacturers. And this viewpoint was shared by many patients, advocacy groups, healthcare practitioners. So in response, the FDA funded 2 major bioequivalence studies comparing generic lamotrigine to the brand name in patients with epilepsy. And the results validated the FDA's bioequivalence standards for anti-epileptic drugs. And in response, the American Epilepsy Society revised their position statement last year, indicating that the FDA standards for bioequivalence for anti-epileptic drugs are appropriate for patients with epilepsy, and that patients and healthcare providers can have reasonable confidence in bioequivalence when you're switching not only from the brand to generic, but also between different generic formulations. So there's different approaches you can take to demonstrate bioequivalence for a product. Traditionally, we relied on in vivo techniques, which comprise of pharmacokinetic, pharmacodynamic, and clinical endpoint studies. But as I alluded to earlier, especially for these more complex drug products, these studies may not be sufficient sometimes, or they just might not be feasible. So we have to investigate alternative methods, in vitro methods, to be able to support demonstration of bioequivalence. There are characterization-based in vitro methods. This is what the FDA calls Q1 and Q2 and Q3 equivalence, which essentially calls for equivalence in the formulation composition as well as the microstructure of the formulation. There's also performance-based tests, which are tests that correlate with and are predictive of human in vivo bioavailability data. Dissolution tests, in vitro permeation tests are also part of this category. Additional reasons for why we need to develop in vitro testing methods. Some clinical studies require very large numbers of subjects, sometimes on the order of thousands, in order to pass bioequivalence statistical limits. And this is because of the high intersubject variability. For products that have modest clinical efficacy, clinical studies just may not be sensitive enough to detect those formulation differences. So alternative approaches are warranted to really provide a more feasible pathway for generic drug approval. In my presentation, I will be defining complex drug products as those products that have complex active ingredients, so things like peptides, products with complex formulations, such as liposomes, iron colloids, those with complex active routes of delivery. This includes products that are locally acting, and also complex dosage forms, so things like transdermal systems, meter dose inhalers. Currently, we have over 260 complex drug products that do not have a single generic counterpart. On the other hand, there's 210 complex drug products that do have a generic, but about 50 of these products relied on in vitro bioequivalence data at the time of approval. So no in vivo studies were conducted for these products. So I generated a plot here to compare the complex drug products that have generics and those that do not have generics. And when you plot this according to the therapeutic area, you can actually see a couple interesting things. So complex drug products that do not have generics, there's actually a wider spread among the different therapeutic categories. So for instance, there's no complex generics for dental use, no implants, no complex generics in the inhalation area, very few parenteral injectable drug products, very few ophthalmic complex generics. The other interesting thing is there's a very large number of complex topical products that do have generics, over 100 actually, but these were approved based on a very well-established clinical endpoint study. And there's a large subset of topical products for which that clinical endpoint study may not apply, or may be a huge challenge to perform that study. So there's about 75 topical complex products that do not have generics. So we would need to come up with alternative approaches in order for us to be able to approve generics in the future. So the FDA has been posting product-specific guidances with alternative approaches, namely in vitro approaches to demonstrate bioequivalence. I've compiled a list here of some of these guidances. We have inhalation products, ophthalmic products, otic products with an in vitro option. We have a number of topical products and also a guidance for a complex parenteral injectable. I've also made this pie chart for approved complex drug products that either do not have a product-specific guidance or do not have a generic approved. So what this would tell you then is you would be able to identify product categories for which we would need to develop bioequivalence recommendations. So you see there's a large number of topical products, inhalation products, intravenous, nasal, ophthalmic products for which we would need to develop bioequivalence recommendations. Taking the same group of products, but now just dividing it up by dosage form, you see that there's a much more even spread among the different dosage forms. So this indicates that our efforts in developing bioequivalence recommendations is pretty equal among the different dosage form types. So we have been focusing our efforts in our research program to really develop in vitro testing methods. We funded 12 extramural projects looking at investigation of key physical chemical properties that affect drug release and bioavailability. We've awarded 15 projects looking at development of in vitro release testing methods and so forth. So lots of significant work that we funded in this area. And if I plot the different projects relating to in vitro testing research and look at it by therapeutic area, we have a pretty even spread. There's a big emphasis on the parenteral products because they have such a wide range in the types of formulations that could be used for this route of administration. So I'd like to give a few examples of research in this area and how the results have impacted regulatory recommendations. The first example comes from meter-dose inhalers known as MDIs. And these devices are used very commonly to deliver drugs for pulmonary disorders. And the formulation, the canister, can be either a solution or a suspension. And what we wanted to see in this study was if you altered slightly the concentration of different excipients in the formulation, would that have an impact on product performance? So we looked at 3 different drug products: albuterol suspension, mometasone suspension, beclomethasone solution. And we altered the particle size distribution for the suspension products, and we changed the concentrations for ethanol and oleic acid. So with these test formulations, we measured the delivered dose. And what we see is that slight changes in the ethanol concentration significantly impacted the delivered dose for the suspension formulations. And then for the solution, slight changes in the oleic acid concentration had a significant impact on the delivered dose. We also measured the fine particle dose, which is the aerosolized particles that have an aerodynamic diameter of less than 5 microns. And we see that many more of the formulation factors impact the fine particle dose. For instance, for the mometasone suspension, drug particle size, ethanol concentration, oleic acid concentration all had a big impact on the fine particle dose. And this is significant because the fine particle dose is the dose that is considered to be actually delivered to the lungs. So this supports FDA's recommendation for MDIs because we ask for Q1, Q2 equivalence, essentially equivalence in formulation composition, because even just slight changes in some of the inactives could potentially change product performance. We also have an extensive research program for ophthalmic products. Here in this study, we worked with the University of Connecticut to formulate different ophthalmic ointments, A, B, and C. And they had the exact same composition. They were actually manufactured with the exact same manufacturing process. The only difference is that petrolatum, which serves as a base for the ointment, it was purchased from 3 different vendors. And we saw that all 3 formulations had different rheological behavior. You see that from the plots for the loss and storage modulus. They also had differences in the viscosity profile. And when we conducted in vitro release testing, we did this in USB apparatus 4 using the semi-solid adapter. All 3 formulations had different release rates. So this indicates that even with Q1, Q2 composition sameness, even by using the same manufacturing process, for certain excipients, if you were to acquire that from different sources, you may have a formulation that has different physical chemical properties, which could potentially impact in vitro and in vivo performance. Late last year, we revised our product-specific guidance on Acyclovir Cream. So initially, our recommendation was for one in vivo study to conduct a clinical endpoint study, but that study is very challenging to perform for this drug product. So we revised it to include an in vitro option. And to qualify for the in vitro option, your formulation has to be Q1, Q2 compositionally the same, and you also have to show equivalence in Q3, so similarity in the microstructure. In order to show similarity in the physical chemical properties for your test product, we recommend a series of tests that you should perform. And I'd like to give an example for how we came up with one of the tests that we recommend the guidance. We ask for measurement of water activity, and this came about through a study that we did with the University of Mississippi, where we measured the solvent activity of Zovirax Acyclovir Cream manufactured in different countries, so US, Austria, and UK. And we also measured it for 2 generic products that are manufactured in Austria. Solvent activity measures the thermodynamic energy status of the solvent in a formulation. So pure water has a solvent activity of 1, and when you move away from that number, the mobility of water or solvent decreases. So for Zovirax, the 3 Zovirax reference formulations, the solvent activities are very close to one another, near 0.7. The 2 generic formulations had a much higher solvent activity of 0.95. And when we conducted drying rate tests after application of the cream on the skin, we also saw that the 2 generic products had a much faster drying rate compared to the 3 reference products. So the implication here is that if a formulation has a very high solvent activity or water activity, then that would increase the concentration of drug in your formulation available for drug absorption and drug release. Okay, the last example I have comes from the research that we do on products that use Plga. This is a biodegradable polymer. FDA has approved 15 drug products that use Plga. I believe the first drug product was approved more than 20 years ago, but we still have not approved a generic product that uses Plga. And part of the challenge is that it's difficult to reverse engineer the reference product and to pick out, accurately understand the key properties of Plga, such as molecular weight, the lactic acid to glycolic acid ratio, and the polymer end cap. So we partnered with a small polymer company to develop a protocol that we actually published about a year ago, where we use GPC and NMR techniques to accurately assay for these characteristics from a marketed product. So we're actually able to separate out the Plga and actually measure these properties. The example that I show here is NMR spectra, which distinguishes between the acid end cap and the ester end cap. So in figure B, there's a slight peak that occurs at around 14 parts per million, and this corresponds to the methyl peak, which constitutes the ester end cap. So by developing protocols like this, it helps generic industry in generic drug development, but also for FDA as we review potential products that come in that utilize PLGA. The Office of Generic Drugs publishes a regulatory science research report each year. So the report for FY16 has been published on our website. I encourage you to go and take a look at this report. It has different subsections, and you can look up the therapeutic area that you have an interest in. This report is linked to our GDUFA website, which I'll show you the link for that in my final slide. And briefly to highlight the new regulatory science enhancements in the GDUFA 2 commitment letter. First, we have committed to conduct an annual public workshop. So this replaces the annual public meeting format that we've had in previous years. The difference is with the workshop now, we have topic-focused sessions. And we also have an open comment hearing period right in the middle of the workshop. So the objective of the workshop, similar to the meeting, the public meeting, is to gather public input to develop our annual list of generic drug science initiatives. And secondly, if industry chooses, industry can form a GDUFA 2 regulatory science working group. The purpose of this working group is to meet with FDA and discuss any concerns or challenges that you face in regulatory science generic drug research. You can meet with FDA twice a year, and we will be posting those meeting minutes publicly on our website. And then thirdly, we've committed to posting annual reports on our website, which detail how all the GDUFA-funded research studies have contributed to development of generic drug products and how they've impacted ANDA review and review of other regulatory submissions. And lastly, I'd like to announce the annual public workshop that we're having this year. So we're actually taking that new format, piloting it this year so that we can roll it out better, hopefully for GDUFA 2. It's going to be next month, Wednesday, May 3rd, at the FDA White Oak campus in Silver Spring. Registration actually closes tomorrow. So I encourage you to go to the GDUFA Regulatory Science webpage, the link that you see here, for registration information. You can attend either in person or by webcast. If you would like to speak during the open comment period, that deadline is tomorrow as well. And again, the purpose of the workshop is to obtain input from industry, any stakeholders from the public, to help us identify what regulatory research areas we should focus on for fiscal year 2018. So in summary, outcomes from GDUFA research studies have contributed to development of guidances and recommendations to industry. And there's an increasing need to develop alternative bioequivalence methods, as you saw, especially for complex drug products. And regulatory science enhancements under GDUFA will allow for greater interaction with industry and the public for feedback on our research initiatives. If you have any questions, please feel free to contact me on my email, our GDUFA Regulatory Science website, which has links to our annual report, as well as information on the public workshop to be held next month, is also at this website. I believe that's it. Thank you very much.
Thank you.